Hi folks, and welcome to another edition of Zog Science. Today we're going to be continuing our discussion about how membranes function in terms of cell transport, and we're going to be talking about active transport. So active transport uses energy to move solutes against their gradients. We talked about in the last video how facilitated diffusion helps to speed up the transport of a solute by providing a passage through the membrane. But facilitated diffusion does not affect the direction of the transport. Basically, it's still moving down the concentration gradient. However, there are some transport proteins that can move solutes against their concentration gradient. Now, in the last video, we mentioned how this moving down the concentration gradient was going with the second law of thermodynamics of increasing the entropy of the universe. Well, because we're doing the opposite in this case, we're going to be decreasing the entropy of the system, and therefore that requires energy in order to happen, and that energy is what makes this active transport. And so we're moving substances against their concentration gradient, and we got to have energy, and cellular energy is usually going to come in the form of ATP. So our passive transport was either the process of diffusion or facilitated diffusion, and that's where we were moving things with the concentration gradient. With active transport, we're moving things against the concentration gradient. And so one of our prime examples of that is the sodium-potassium pump, where we are pumping sodiums into the cell and we are pumping potassiums out of the cell. And in both cases, we are moving things against their concentration gradients. Other examples of this are our proton pumps, where we pump protons from one side of a membrane to another. Again, we're moving things against the concentration gradient. We can also have co-transport. This is when active transport happens through indirectly moving one molecule that then moves another one. This is something that plants do a lot. They use proton pumps to drive active transport of nutrients into the cells. So for example, this sucrose co-transporter, what happens is that the proton moves into the cell through the co-transporter, and when it does so, it also moves in the sucrose molecule with it. So it's co-transporting it. Now, the examples we just looked at are where we move specific small solutes across the membrane, but we can also move larger molecules. And so these occur by bulk means, and these bulk means require energy, and these can be either in or out of the cell. So exocytosis is the first one. This is where we move things out. A transport vesicle, which we'll learn a little bit more about it later in the unit, is going to migrate to the membrane, fuse with it, and release the contents. And many secretory or secretory cells use exocytosis to transport their products. So they make something, and then they transport them to the outside of them using exocytosis. With endocytosis, the cell takes in molecules and particulate matter by forming new vesicles from the plasma membrane. And this is a way that a lot of single-celled organisms will consume other organisms. So for example, amoebas will eat other cells by engulfing them and bringing them in through endocytosis. There are 3 different types of this. You're not going to be expected to really know the 3 individual types, but I wanted to make sure that you at least saw them. So phagocytosis is bringing in large amounts of things. Penocytosis is bringing in a little bit of things, and then we have receptor-mediated endocytosis. So phagocytosis, bringing in a large molecule and sucking it in. Penocytosis, just a little bit of something and bringing it in. And then you have receptor-mediated endocytosis. This is where the solute that you want to bring in is going to bind to a receptor molecule and one of those proteins, a protein, probably an integral protein across the cell membrane. It's going to bind, and that is going to cause the cell to then begin an endocytosis movement to bring the food into their cells. Again, not super important that you know the specifics there, but make sure that you understand the general concepts of exocytosis and endocytosis. All right, that's all we've got for you today about active transport. Thanks for watching, and we'll see you next time.